Hi, my name is Carol and I am a Lycra Solutions and Support Scientist. Today, we're going to be talking about the best practices on how to image your chemiluminescent Western blot membranes. But first, let me tell you a story that happened to me a few years back when I was in the lab. So I had to silence this DNA repair gene. So I went ahead and I treated my cells with some siRNA. I allowed them enough time to do their thing. I collected the cells, I extracted the proteins, and I ran a blot. My goal at that point was simply to verify that the levels of my protein were significantly reduced. And I was thrilled to see that I had 85% silencing. Of course, I had to repeat that data, so I used the same proteins, I ran virtually the same Western blot protocol, and that's when I wasn't so happy because I got only 60% silencing. Well, which data was correct? 85% silencing, 60% silencing. What should I do at that point? So I spoke to several researchers and Western blot specialists, and it took us a while, but we finally determined that the problem was that I wasn't capturing that enzymatic reaction at the same point every time. So I paused everything, I went back, optimized and standardized my protocol, and boom, reproducibility was back, and I was able to get the consistent data that I needed. So what that story tells us is that while there are a lot of intrinsic variables to a Western blot protocol, such as the fact that we have to extract proteins and transfer them to a membrane, when it comes to a ChemiBlot, there's a whole additional layer of variables that we have to deal with. Those variables come in place when we're trying to image that blot because we're going to be depending on the signal produced by that enzymatic reaction that happens between the enzyme that is conjugated to our secondary antibody and the substrate that we add to the membrane. That enzymatic reaction is going to be varying with several different factors. For example, time, temperature, the amount of substrate, the type of substrate, and so on. So our main goal when performing a chemiluminescent Western blot is to try to reduce and control these variables as much as we can. So let's take a look at this graph to have a good understanding of what happens with that enzymatic reaction through time. So on this graph, on the x-axis, we have time as a variable, and on the y-axis, we have signal intensity as a variable. Once that enzymatic reaction starts taking place, we see what's on the left of the graph, which is called the tow region, which is what we observe when that reaction is just starting to take place and we don't have enough signal to try and capture and quantify it accurately. On the other hand, if we let that reaction go on for too long, that enzymatic reaction reaches a plateau, which we can observe on the right side of the graph in what we call the shoulder region of the graph. Again, at that point, we cannot accurately capture and quantify the photons of light produced by that enzymatic reaction. So the bottom line is, if we want to be quantitative with our ChemiBlots, we need to capture that reaction right here in the middle range of that graph. Now let's take a look at what would happen if instead of using a substrate that was warmed up to room temperature, I used a cold substrate instead. See how much that curve has shifted? And that is only one of the variables that we have to deal with. Hopefully, these graphs have shown you how much an enzymatic reaction varies through time and how much those different factors can influence the rate and curvature of that enzymatic reaction. That means that it's going to be extremely important for us to properly plan and perform our protocol during our chemiluminescent Western blots.